Hello, thanks join my webinar, and I also thank GenScript to support this webinar. Today I'm happy to share our recent projects on COVID-19 drug repurposing. As you know, SARS-CoV-2 is dangerous virus, and you are now seeing the daily death of the patient diagnosed with coronavirus. The number of the daily death around the world is around 400 when it first outbreak at early last year, and actually we are now just past the second peak, but actually very recently the number is a little bit increasing again, so this is kind of concern. The number at the end of this March is around 10,000, so this actually shows that we definitely need some kind of rapid response to cure this disease. Of course, vaccine is one of the really good way to do, but we definitely need therapies to cure this disease. So one of the way we can deliver this therapy to the patient is using the drug repurposing strategy rather than the complete new drug discovery. The reason is, as you know, the new drug discovery takes more than 10 years, but in contrast, the drug repurposing usually takes much shorter time, and we can use some kind of cutting-edge technologies, including machine learning or AI or some other really advanced molecular biologies. Then we probably reduce this time a lot than before, so that we can deliver this therapy to the patient in the right time. So this is one of the main reasons that why we took drug repurposing as our main strategy for this project. The SARS-CoV-2 virus itself is not really dangerous, but with the real dangers come after its rapid replication by hijacking our host system. And actually it only takes 1 or 2 days, and after this this virus actually triggers our innate immune system in 3 to 5 days, and eventually that causes cytokine storm or other severe symptoms in a week time. So there might be two main opportunities for us to cure this disease. One of them is like early stage intervention to block the virus replication, so that it couldn't cause any kind of severe symptoms afterwards. And another one is like if the patient already in a later stage, then maybe we can using some kind of immune suppressor or other way to cure this disease. There has been huge effort in the scientific communities to understand this virus and the disease, and many papers submitted to the preprinting server and also the peer-reviewed journal, and kind of the more than half of the preprints are now in the medRxiv and SSRN, and in particular in the medRxiv, I think the other journals or preprint servers are more or less same, but in there kind of more than 2/3 of the preprints are related to COVID-19. This clearly shows the kind of the trend and also the efforts in the scientific communities for COVID-19. This is one of those papers where they reported the directly interacting proteins to SARS-CoV-2. As you know, SARS-CoV-2 has its own structure, and actually it does have quite large size genome, and there are subgenomic structure of those viruses, and there are non-structural proteins and also structural proteins and also open reading frames. So we now know their structure, and this paper used mass spec to measure and identify the directly interacting protein in our human body. And these red diamonds at the center is the subgenomic structure of SARS-CoV-2, and those nodes are the directly interacting proteins in our body, and they reported 332 proteins as a deep protein. So we used this data set as one of our data. The second data we used is DEP proteins, the differentially expressed proteins. So this is another paper, and they also used mass spec, but actually they measure these expression changes at 2, 6, 10, and 24 hours after infection. And this is the summary of the results. And as you can see, there are certain different patterns or changes over the time. In particular, 6 and 24 hours shows some significant changes at the protein expression levels. So we used this DEP data as one of our main data sets. So Woo-Chang and Maeve in my lab, they work on these projects, and we have made a hypothesis that once the SARS-CoV-2 virus comes into a body that connected to or interact to the directly interacting proteins, and then eventually somehow that controls and regulate some kind of expression changes of the DEP proteins. But there were questions like how this DEP protein actually controls these changes. So we thought there must be a hidden layer in between the DEP and the DEP proteins. So we decided to construct this hidden layer. This is protein-protein interaction network, and then we got a really huge network with so many different proteins. So we definitely need some more systematic way to identify or analyze this network. So we use network algorithm to uncover key pathways in this hidden layer. And then the real kind of question is what kind of approved drugs actually can effectively target those key pathways. So we used drug simulations to identify these approved drugs. So this project's actually available in bioRxiv as a preprint. So if you have any more interest, or if you wanted to see more details, please read our preprint. And this is in revision in a peer-reviewed journal, so hopefully it will come out very soon. So after the network construction, we called it a SIP network, the SARS-CoV-2 induced protein network. So in this SIP network, we have around 13,000 node proteins, and also 344 different interactions at 6 hours, and at 24 hours the number is slightly higher, but it's more or less same. So it does have the deep proteins in pink and the DEP protein in blue, and in between there are hidden layers. So this is the real network. And as I said, after construct this network, we tried to identify the key pathways in this huge network by utilizing some network algorithms. And in particular, we're using 4 different network algorithms that you can see here: random with restart, eigenvector, degree, or between essentialities. So this is the algorithm, and I would like to quickly introduce why we used it or what it actually tells us. So this is the network, the protein-protein interaction network, and this already looks quite complicated and complex, but actually there are only 164 proteins in this network, but it's already very complex. But as I said before, we are dealing with more than a couple of thousand protein network. So the complexity will be really, really high with those higher number of proteins. So what we actually used is the kind of different algorithm, but this is the random walk with restart. And actually what it gives us at the end of the day is like the score per each node, the score per each proteins. So these scores giving the information that whether this is particular protein is more important than the other, or this is less important than the other. So we can make a rank to understand which protein is most important in this network and why. So this looks very complicated, but actually what this kind of algorithm being used in recently in other engineering area is here, like Netflix. I think everybody likes Netflix, and they have kind of very interesting function that they recommend new movies for you because you watched movie XYZ. How they do? They're using network analysis. Also, in LinkedIn or Facebook, they often recommend you a new friends or new connections. How they do? Again, they're using the network analysis. And also in shopping, particularly Amazon, they giving some kind of emails and some recommendation to giving you some kind of new list of shopping items, and they say you may also like those kind of items or products. How they do is like they using the movie or your friends or your purchased kind of items in each node in the network, and then they run the network analysis or algorithm to identify most promising new movies or new connections or new product for you. But in our case, we're using the proteins in each node. That's the only difference. But in the end, it tells us some kind of the relationship with some kind of unknown node, unknown protein, or unknown connection, unknown movies, and that could be very informative and important. So after running this algorithm, we got this kind of more informative figure. Actually, this is exactly the same as the upper part of the network, and I just highlighted the important nodes and important interactions in between two proteins. So the color red means this is highly upregulated in certain disease, and this blue is downregulated in certain disease. So actually this is quite helpful and powerful. Just to give you a little bit more information about network algorithms we used in this project, there are 4 algorithms, and actually the reason we're using these 4 different algorithms is that the 4 different algorithms actually giving different informations. So random with restart is really good to understand which node is most important and having a closer relationship with a specific node that's usually the protein of our interest. So this is really powerful to identify those proteins. But there are some other kind of important proteins in the network. One of them is maybe this kind of proteins that is not really close to the protein of interest, but this one is actually bridging or connecting two subnetwork. If there is no protein like this, and then in the network these two subnetwork couldn't be connected well. So this kind of protein can be identified by this between the centrality algorithm, and also degree of centrality is the one that identifies half protein in a distal subnetwork. So each algorithm are having different functions, and that's the reason we use these multiple algorithms. And this is just a slide that shows the example after running this algorithm, what sort of the proteins could be identified in each algorithm. In random walk, as I showed in the previous slides. It usually identify the close by the protein of our protein of interest like this, but like between the centrality, our protein of interest here, but actually the identified one by this algorithm is like something that connecting some other subnetworks, and also degree of centrality as well. It tells us some different information. So this is the examples of the algorithms. So let's come back to our story, and this is the results of our network analysis on the SIP network, and we identified 238 proteins that are common in between 6 and 24 hours, but actually there are certain number of proteins at 6 and 24 hours which is very specific for each time point. So this is one of the kind of summary for the network analysis results. And also there are another overlapping analysis to show where these key proteins are located in the SIP network. So as you remember, there are deep protein which directly interact to the SARS-CoV-2, and the depth proteins which are changing their expression after the infection, and there are hidden layers. And actually this kind of purple or blue color region is actually the one located in the hidden layer. So actually there are certain overlap between this key protein and the deep, meaning quite many of them are actually deep proteins that identify key proteins, but actually there are many proteins sitting in the hidden layer, as we hypothesized in the earlier stage. So this is the kind of information. And also what we did was we checked the disease enrichment of our key proteins at 2 different time points. You are now seeing these top 10 ranked disease names in each time point, and as you can see, particularly the red ones are the ones that are known to be related to the symptom of the COVID-19. So we think we identified good subset of key proteins using the network analysis. And also we did another quick tests with network, which is here. So we just visualized the interaction between the key proteins, and as you can see, they are highly crosstalk to each other, which is good sign. And in the common and also at 6-hour specific networks, actually there are quite broad terms, and this is quite diverse. But actually one thing is very interesting at 24-hour specific protein network, which are here, particularly in here, like ribosomes and the infection related and RNA virus related, so on. So it seems like 24-hour specific proteins are more related to maybe the virus replication. So as I showed before, this is the SIP network, and we tried to highlight the important connections and the important proteins in SIP network by utilizing these network algorithms. But actually this is not really informative, so we decided to change the way to visualize this network. So what we did first is like we wanted to divide the proteins and protein interactions by the 3 main categories: the deep, depth, and the hidden layer in between deep and depth. So there are 3 main categories, and also as I said before, the SARS-CoV-2 does have the subgenomic structure, so we subdivided the deep categories into the subgenomic structure, And also in the hidden layer, we're using the many different pathway databases and others to identify the functions of the protein in hidden layer, and we divide by three main groups. One is immune system and cell cycle and metabolism of RNA and some other functions. So this is the subcategory of each three main groups. And then also we did one additional analysis of the protein, which is subcellular localization study. So we wanted to know where these proteins are located in our cell. There are many different places like nucleus and ER and Golgi, and we put them in a different color, like here, and this is in the inner circle and here. And the last one is the most important information, which is the interaction. So we put that in different colors. So for example, if the interaction is with a deep protein, then it should be in orange. If this is with that protein, that should be in green. And also that follows to the other subcategories of hidden layers. This is one of the alternative way to visualize a network. This new visualization is more informative, but actually if we compare one of this visualization to the other, will be more informative. So the one I showed you in previous slide is the network for 6 hours. But if we compare the 6 hour with the 24-hour network, then it's quite useful. So as you can see here, and maybe you already spotted, there are kind of huge increased interactions in between the deep and the metabolism RNA, and also the nucleocapsid protein to the metabolism RNA. And also another thing is like the internal or inter connection in the hidden layer from or to the metabolism RNA is hugely increased, as you can see here. But in here, there is not much interaction at 6 hours. So there are many things to discuss, but this is, I think, one of the key messages in this comparison. And as you know, the NSP8 of SARS-CoV-2 is already known to having a key role in the virus replication and also the nucleocapsid as well. So this is one of the most interesting finding in this analysis. And this is not just by eye. It's because we also checked their biological function enrichment, and as you can see, there is a certain enrichment terms at 24 hours. So the 6 hours are in the blue color, and in compared to these 24 hours, there are really what can I say, less enrichment at this particular terms. And what are those terms in the far left side is here. And as you can see, they are related to viral process or transcription, and also some kind of RNA related functions. So it clearly shows that at 24 hours, many things going on the virus replication. So next, we wanted to understand and identify what are the kind of the drugs that effectively target those proteins. So we identify those proteins in the network, and then we brought in the approved drug database where we can find nearly 2,000 approved drugs, and then we used network proximity measure techniques to simulate and identify what kind of approved drugs could be useful to inhibit those specific protein or pathway. So we ran this proximity algorithm for all each compounds in this database. After this drug simulation, we identified 200 of approved drugs for COVID-19 drug repurposing, and 40 out of 200 identified drugs are already in COVID-19 clinical trials. And 34 drugs are already reported in COVID-19 research. So in total, 70 out of 200 are already related to COVID-19 trials, or they are already being reported. So this somehow kind of show the validity of our approach. And here are some examples of the drugs. So some of them are chloroquines and dexamethasone, which is quite well-known drugs already, and these are also identified in our approach, and some others. So this is just example of the 40 drugs that are already in clinical trials. And also we checked the therapeutic areas of the 200 drugs. Just wanted to know and understand which therapeutic areas those 200 drugs being approved by the regulators. Interestingly, but not really surprisingly, the cancer therapies are the top. So they are around 1/4 of the 200 drugs cancer, related cancer, and the other one is like, second one is the hormone related and also estrogen related ones, and the third one is about the diabetes. So this is just like some additional information about 200 drugs, and more interesting bit is in here. Often a drug approved in one specific therapeutic area, but is also being tested or trialed in some other therapeutic areas. So we checked what kind of clinical trials are also going on for those 200 drugs. And interestingly, nearly 35% of the 200 drugs are being in clinical trial that related to infectious disease, and also around 18% are related to HIV or inflammatory and respiratory disease. So this is one of the interesting information that we found in the analysis. After this, we were really curious about what could be the mechanism of action of those 200 drugs. So we further studied about the association or relationship between the 200 drugs and the key pathways. So this is just shows the summary of the relationship. As you can see, the y-axis showing the 200 drugs, and the x-axis showing the 148 pathways. And there are some kind of the relationship, and some are quite specific. But in this way of the kind of simple clustering, it's quite difficult to see the pattern and understand or analyze this. So we decided to using some more advanced techniques. So to do some kind of more advanced like machine learning type analysis, we need some kind of sophisticated data. So in here you are thinking the kind of just like a conceptual kind of video, but we done kind of drug pathway association study by checking all the drugs are related to what type of pathway so far so on, and then actually we made one large matrix that contains the drug pathway association, and then we put this as a kind of training input for the artificial neural network, and we train the neurons. So in here on the right upper corner, you can see the artificial neurons, and maybe it's too small, but there are, so it's difficult to see, but there are kind of gray lines in between the neurons that's connected each neurons. And on the left bottom corner, there are red kind of cross, and those are the data points that's directly coming from this pathway drug pathway association matrix. So this actually trained 2,000 times by iteration, and as you can see, after the training, each training, these artificial neurons are now spreading over the data space to capture all the data points. In particular, there are two main data cluster in here and there, and then these neurons are actually taking more space in those particular cluster, but actually some other neurons are spread over to some other data points to understand and capture all the data in this association matrix. The artificial neural network analysis provided this result, which is 148 heat maps for 148 key pathways, meaning each heat map represents each pathway, and actually the heat map consists of the neuron, artificial trained neurons, and the neuron in each heat map has the same kind of location. Like if a specific neuron is in the top left hand corner, and this neuron must be in the same place in each other neurons across the 148 heat maps. So that allows us to directly compare, visually compare each heat map easily. So for example, if you can see here, there are kind of 4 or 5 heat maps that has a strong signal at the right bottom corner. So if you can have a closer look on the name of this pathway, actually it's not up here, but if you are interested, please go to the bioarchive and have a look on our preprint. All the information is available, and those are all related to toll-like receptor pathway. If there is kind of signal like here on the left bottom corner, they do have a really visually similar pattern, then those heat maps and the pathways are quite related. It's because they are all cell cycle related pathways. And also there are another example, like there are 12 o'clock or right upper corner. If there is a kind of strong signal, then those are related to the metabolism of the lipid process. So this is the kind of heat maps that we get after the artificial neural network analysis, and this will eventually merged and clustered to get some more data. So 148 key pathways are merged in this U matrix, and then actually after using number of different clustering algorithm, we successfully identify 9 main key pathway clusters, and the name of clusters are here. As you can see, the metabolism of RNA and protein and DNA replication and cell cycle, and also some cytokine signaling and toll-like receptor signaling. So it's quite related to the immune system. So as you can see in here, like greenish parts are more or less the immune related ones, and those blue and yellows are the ones that more related to viral replication. So after this clustering, we identified 2 main mechanism of action categories. One is virus replication, the other one is the immune response. And also the artificial neural network gives more information about the drug relationship to each neurons. So at the moment there are numbers inside of each neurons here, and this 13, for example, these 13 drugs are strongly related to the pathway showing the signal at this place, and so forth, so on. So we can actually have the list of drugs that are related to what type of mechanism of action by this analysis. So this is actually the name of the drugs that belong to each neuron and each pathway and each mechanism of actions. Sorry for the small font. Again, if you are interested, please visit the bioarchive and our preprints and have a look in a large font size. And this is the final kind of analysis of the artificial neural network, and the reason we choose those here, like if we see the life cycle of all virus replication mechanism of the SARS-CoV-2, once the SARS-CoV-2 comes into our body and get into our cell, it actually goes to many different stages to replicate the virus itself. And then what we think is, like, this is quite interesting. In certain point, like the spike protein or envelope protein generation, actually metabolism repeat function is really important in ER, and also in the RNA replication process itself, definitely the RNA polymerase function or metabolism of nucleotide or cell cycle related to pathways are really important. I think this is why we categorized the 9 clusters into each of 2 mechanism of action categories. We also wanted to know that what proteins are directly targeted by the 200 drugs, and whether there are any kind of commonly targeted proteins by multiple proteins. So this is the results, and actually there are 30 proteins that are targeted by more than 8 different drugs, and this is the quick network diagram of those 30 proteins, which are highly connected to each other, and they do have very interesting functions. So we checked their most enriched function of those 30 proteins, and as you can see, the top ones are here, and those are strongly related to the nitric oxide generation or NADP generations. So after this, we having around 15 drugs that are specifically targeting those functions, and after filtering out with a number of safety issues, we only having 5 drugs at the time that could be very promising for the repurposing for COVID-19. So we decided to experimentally validate those 5 different drugs in the cellular assays. To validate our hypothesis, we collaborated with Friedman Weber's lab in Gießen, Germany, and they do have 2 different cells, VIR-6 and CALO3, and also they did have the SARS-CoV-2 virus. So we tested on the VIR-6 first, and then those 2 are the controls, and those are 5 drugs. And as you can see, 3 drugs of 5 are not really showing good efficacy, but actually Proguanil and Sulfasalazine showing very good efficacy in the VIR-6 cell line. And also we checked the cell viabilities, and actually we couldn't see any kind of huge issues on this, so we were quite happy about this. And then we wanted to check on the CALO3 cell line, and also we wanted to check the dose response of those two drugs that shows really good efficacy in the first validation. So in Proguanil and Sulfasalazine, as you can see, actually we also have the CALO3 data, but in here VIR-6 it really shows really good dose response, and also in CALO3 we definitely have really good efficacy in here. In further, we collaborate with Costas, who is in the Miller Institute in Cambridge, who help us to understand the biomarker studies, which published by Krogan group in Cell, and they reported a number of important biomarkers of COVID-19. Two of them are CXCL3 and IFNMB1, and also we tested in color 3 and VLO6. So more data is available again in preprint, but in here we are showing only color 3, and we confirmed those two drugs actually showing a good efficacy on those two biomarkers. So we studied a little bit more about what makes the difference in between the 5 drugs. So our kind of hypothesis is sulfasalazine and proguanil actually targeting 2 different main pathways for nitric oxide generation. One is the nitric oxide generation itself, like NOS related ones, and also there is another one like NADP generation process. So sulfasalazine and proguanil, as I said again, these 2 drugs targeting these 2 different pathways at the same time. Actually, the 3 drugs that didn't show the good efficacy is targeting only one pathways at a time. I think maybe that could be the main difference. So we just finished the paper with this diagram. So in summary, we started with 2 different main data sets to get the 332D proteins and the DAP proteins at 6 and 24 hours. And then we constructed the network in between the DIP and the DAP protein to having the hidden layer at 6 and 24 hours, of course. And then we ran 4 different network algorithms to identify the key proteins and the key pathways. And actually we identified 320 at 6 hours and 394 at 24 hours, and there are certain commonalities, but also there are important specificities for each path at each time point. After this identification of key proteins, we ran network proximity measure to identify certain set of approved drugs for COVID-19 repurposing. So we tested 1,917 approved drugs in a database, and then actually we successfully identified 200 approved drugs. After this, we did further analysis for the MOA, and then we identified 148 pathways are related to those 200 drugs, and then we successfully clustered 148 pathways into 9 main groups, and then that led us to identify 2 main categories for mechanism of action. One is virus replication, and the other one is the immune response. Finally, we shortlisted 5 drugs that related to the virus replication with NO production, and then we did wet lab validation on 2 different cell lines, VLO6 and CAL3, and finally we confirmed the efficacy, particularly with the nice dose response curve and also biomarker inhibitions with 2 approved drugs, Proguanil and Sulfasalazine. So we hope this data could be useful for the further research and analysis and possible therapy development. So this is it, and I would like thanks to my group, in particular Uchang and Maeve, who led this project, and also my fellow group leaders who leading the experimental groups in the Miller, in particular Costas, who help us to validate the biomarkers and the experimental validation, and the other collaborators in Cambridge and elsewhere in the world, particularly Fredman Weber, who help us to validate our hypothesis on the bench, and the funders, in particular LifeArc, who are helping us to do this research. So thanks for your attention, and I'm very happy to take any questions. Thank you.